This is going to be a very different talk. And I'm going to try to convince you by the end of this 10 minutes that maybe viruses are worthwhile considering. Okay? And I guess within the last 2 years, you have been convinced about the importance of at least one virus. But there are others as well. And there are even some stuck in our genome that we'll talk about. So, but anyhow, that's been my career. I study viruses that affect the central nervous system. Started first with the AIDS pandemic, and then there have been one after another. It's been Ebola or Zika and the coronaviruses. So, so sorry for the people who are online. I guess you'll have to do without the slides. You'll have to imagine what's being presented. So I'm in the intramural program within NIH, and this is the Bethesda campus. Wish we could have actually held that meeting there. You would have really liked it. But these are good venues as well. So let's see. Let's go to full screen here. Oops. And now, yeah, okay, this works. Okay, so I've divided my talk into 3 parts. The first is effect of viruses on neurodegenerative diseases. The second is that viral infections themselves can have a very specific neurodegenerative syndrome. And lastly, viral infections could potentially be a cause of neurodegenerative diseases. So the first part, that is effect of viruses on neurodegenerative diseases. And I'm going to just present some examples of each. There's no way to do an exhaustive talk on these things. And I wanted to start with SARS, since everybody is now an expert on coronaviruses. And so can SARS precipitate Alzheimer's or accelerate Alzheimer's and Parkinson's disease pathology? That's what we've been worried about. We don't really know the answer. But there are little bits coming out in the literature suggesting that it's a reasonable thing that we need to at least follow these populations and see what's happening. The first one on top is a Simian model in which these animals were infected with SARS-CoV-2, stays in the lung, doesn't go into the brain. But what they show is that there's a whole host of synuclein pathology that you can see in the brains of these animals. The second at the bottom are these graphs showing you of individuals with SARS-CoV-2, and they're following them with serial CSF analyses or blood analyses of A-beta peptide, and showing you that there's a decrease in these A-beta peptides in these individuals. However, these neurofilament levels are actually going up, suggesting that maybe some AD-like pathology may be in the early stages of this disease, right? So what about viral infections? Can they cause neurodegenerative syndromes? And I've got a whole host of these things. Typically, we think about viral infections causing an acute encephalitis, and after that, patients get better, or they don't, and that's what you're left as a sequelae. But that's not really true. What we do have is there are a number of viruses that can cause chronic illnesses. So you have poliomyelitis, can cause a post-polio syndrome. Zika, as you know, can cause congenital Zika syndrome. They'll infect the brain directly. Measles can cause a subacute sclerosing panencephalitis. Again, you'll never find virus in the CSF, but the brain itself is loaded with viral proteins. HIV causes a dementia. And then there are others. Dengue can also cause a progressive dementia, and the Spanish flu that we've all learned about from the... Oh, you can't hear it? That's one thing my mother never taught me. Okay, and the Spanish flu, which we know of in the early 1900s, where individuals were left with encephalitis lethargica following that. Just to remind you about the post-polio syndrome, it's still real today. There are very few autopsy studies that are available on these individuals, and the few here show that there is persistent virus in the spinal cord with inflammation around it at the time of death. And that's with an incubation period of several decades, right? from when they develop polio. What about HIV? HIV can cause a dementia. It's a subcortical dementia. So as you can see on this MRI scan here, this is a 30-year-old individual, and there's a loss of...
Sorry to interrupt again. I need to get you connected in there. I don't think you're sharing the screen in this Zoom.
Yeah, we tried that and didn't work. But if you have a mechanism of doing that, that'd be great. You got it? Oh, great, man. That was easier than I thought. Okay, so you can see it? Okay, great. Okay, so that's the MRI, and then right next to it, you see the pathology on top. I don't know if you can, actually, let's see if the pointer does work. Yeah, here you go. At least you can see the pointer. So these are perivascular cells, mainly macrophages, that are infected with HIV. So you can see the TAT protein, GP120, and astrocytes that have a restricted viral replication. And I want to bring up this concept of restricted viral replication, which is very important. That means the complete viral particle is never formed. What you do have is some viral proteins being formed, but those alone are sufficient to drive the pathology of the disease, right? And if you look at neurons, they degenerate, but they're never infected, okay? So before that, we always thought a virus infects a neuron, it kills it, or rabies, herpes, whatever, and that's how the disease pathology occurs. What we realized is that in most viral encephalitides, you see a lot of neuronal loss, but the neurons themselves are almost never infected, or very few are, okay? So you have to envision some kind of an indirect mechanism, and the indirect mechanism okay. So what we think at least is happening here is that you have these infected microglial cells / macrophages and astrocytes. They produce viral proteins, which we've termed viral toxins. They can then activate other resident glial cells or contribute to neurotoxicity either directly or by all kinds of other things that are being produced by activated glial cells. Right? If you look at the pathology of HIV patients, again, it's very complicated. Okay? It's complicated because you see a lot of other kinds of pathology mixed in there. These are individuals who died at these different decades of life, and you can see that amyloid plaques are increased at every decade of life compared to controls. Okay? Now, you pointed out different types of amyloid. Here, these amyloid are somewhat diffuse plaques, right? But that's what you see most of the times in these individuals. So the question is, how can they be formed? And so there are many different mechanisms. Here we've shown that the TAT protein, even if you put patients on antiretroviral drugs, right, and you control viral replication, TAT is still being produced and released into the CSF. So you can see that it can activate beta secretase. So here you can see that on this thing. And by doing so, it can increase amyloid beta production. It can also inhibit TAT uptake and can also inhibit neprilysin here in a dose-responsive manner. So it'll prevent the breakdown of A beta. So the cumulative effect of this is aggregation of TAT and amyloid. So this is a transgenic mouse where we induce TAT because the doxycycline inducible promoter there. And you can see extracellular aggregation of TAT and amyloid in the brain. So we then did atomic force microscopy. And as we put TAT in there in increasing amounts, you can see on these A beta fibrils, they tend to accumulate here. It decorates the fibril. And then later, it forms much larger plaques over here. And these are neurotoxic. Okay, so now what about infections, viral infections as a cause of neurodegenerative diseases? And here I'm going to point to there's a lot of interest in the possibility of herpes simplex virus in Alzheimer's disease. And then we've been interested in the possibility that endogenous retroviruses may be important in ALS. And just recently, there's been a resurgence of interest in Epstein-Barr virus in multiple sclerosis. Okay. So I'm going to talk only about endogenous retroviruses today. And just to tell you as to if you look at our human genome, there are more retroviral genes there than there are protein coding genes. So when most people say, well, I did whole genome sequencing, they really only sequence 1 to 3% of the genome. That's what they're looking at. What they're really talking about is a whole genome sequencing. There's a huge hole in the thing. About 8% of the human genome is retroviruses, okay? And about 40% of the human genome are transposable elements. So they're all retroviral sequences that we've acquired through the process of evolution. And what separates us from chimps is really the acquisition of this one endogenous retrovirus, which is HERV-K. And there are about 100 copies of that in the human genome. Not much has been studied in the context of neurodegenerative diseases, but they are being implicated in several diseases. And there are just few papers here and there suggesting that possibility. And I'm going to tell you a little bit about the ALS part of it. So as most of you know, it's a devastating disease. And about mortality is about 90% by 5 years. And most of it is sporadic. And what is fascinating about the disease is the anatomical spread. So, and it spreads very rapidly. Somebody's got to explain how the disease actually spreads from cell to cell. So we looked at this one endogenous retrovirus called HERV-K that I told you separates humans from chimps. And what we found was that you can find that there's large amounts of reverse transcriptase that you can find by immunostaining here. This is the envelope in the pyramidal neurons here. While in the controls, we never found it. And we also find it in the anterior horn cells, but not in the lateral horn or the posterior horn of the spinal cord. We looked at the RNA-seq data from the New York Genome Center. And you can find these different loci where there's each dot represents a different individual. And you can see clearly in ALS patients, there is activation of these loci where the HERV-K reside. We made a transgenic mouse model, and what you can see is that here we force the expression through a Thy1 promoter, so it's being expressed in neurons. But if you didn't know that this was a mouse, you would say it has ALS. It develops classical upper and motor neuron signs, even though the protein is expressed in all neurons. But nothing else degenerates, okay? So you can see both here involvement of the lower motor neuron and the upper motor neuron with degeneration over here. And you can also see its expression in the anterior horn cells, and the remaining neurons that are left there become really large because they're re-innervating the muscle. We then screened a whole host of antiretroviral drugs against HERV-K subtype HML2 in culture, and we found that the antiretroviral drugs have some activity, but they're not that great. If you compare them to HIV, for example, you'll require 3 times the amount of AZT before it does anything. To our surprise, there was one only, which is a Becauer, that actually seemed to work better with HERV-K compared to HIV. But all the others really are not that great. Nonetheless, the group in Australia did a open-label clinical trial on 40 patients. And we asked them to send us blood samples. And we have an assay that we've developed for HERV-K in blood. And we found that at about 24 weeks, the levels actually did come down. So it took a long time for levels to come down. When they went off the drug, there was a very quick rebound. Okay? But when we looked at the individuals whose levels came down, we found that those individuals had lower levels. Their disease duration was also shorter. Okay? And those individuals who did respond, their ALS rating scale also, they did go down, but not as rapidly as those who were the non-responders. Okay? So it seems to suggest, at least in an open-label study, that is worthy of pursuing further. So I'll stop here and acknowledge the people who actually do the work in my lab. These are the other collaborators here, including the work on TAT that I showed you was done in conjunction with Eliezer Maslier here. Stop here and take questions.
Very thought-provoking. We only have time for one question now, but write your question down for those of you I don't get to because there's a discussion period. You were first.
Thank you for the very interesting talk. I'm wondering whether you have examined the relationship between that ERV expression and TDP43 pathology, and also whether you have examined some other types of transposons like line 1 elements in this context.
Yeah, so yes, so in our transgenic mouse model, you will get cytoplasmic aggregation of TDP43, right? But it takes a good about 10 months before that happens. So about 10-month-old animals will develop TDP43 aggregation in the cytoplasm. Now, we have not looked at line 1, but some other groups have. And yes, you see line 1 activation in a lot of diseases. So I don't know really what to make of it. But yes, you do get some line 1 activation.
Thank you. That was
Sure.
fascinating.
All right, thank you.